So I'll just spend a couple of minutes doing this, I want to finish up. I'm going to spend a couple of minutes doing this. So what we've done in some recent experiments is to record from these neurons, the ones that in HVC that project to that anterior pathway, the variance injecting pathway, the one that evaluates errors during singing. And what we found is something quite remarkable in this group of cells. And perhaps there's been no other observation like this, or at least as compelling as this, in any other study so far. So we know what the cells look like. We can be very confident we're recording from them. So here is a result that may not seem so remarkable to you, but if you know the neurobiological literature, you would see this as being quite surprising. So here are recordings from one bird, red 240. This is now an in vitro preparation. We've got a thin slice of tissue, brain tissue, that's alive. And because we've arranged it that way, we're able to do intracellular recordings much more efficiently than we could if the animal was alive and intact. So we can record from lots of cells, but obviously that slice is no longer, that bird is dead, is no longer singing. So there are limitations to what we can do, to be sure. And this might look to you like a bunch of traces from one cell, from red 240, but that's not true. Each one of these traces comes from a different cell. But they all look pretty similar to each other, right? And each one of these traces come from different cells in blue 88, and they look very similar to each other, but they look different from red 240, like the timing of the spikes, for instance. The same thing for this bird here. That's not how the system's supposed to be organized. Nobody ever thought there would be such organization, and statistically we can show that these are highly significant variation. If we now look at the actual shape of the spike waveform, which in principle shouldn't be carrying any information, we don't think about it that way, except for, well, I'll get to it in a moment. Here are now, for 10 cells in a different bird, overlaid spikes. And they all look really similar to each other. Here are 10 or 9 overlaid spikes from another bird, look very similar and different from the first one, and so on and so forth. Okay? So something's going on there, which is a lack of variation and a source of variation, which is quite different than what one would normally expect. And again, the statistics for the first spike variation is highly significant. So how do we deal with this? Well, I caught myself from saying something silly, which is, of course, there's a relationship between the spike waveform and the bursting or the activity of the cell. That's what we talked about at the end of the last lecture, right? It's the Hodgkin-Huxley equation. It's the information that the cell transmits from its inputs to its outputs via the activity of these various ion and calcium-gated channels. If those features are varying from a cell-to-cell basis and from a bird-to-bird basis, those are features of the intrinsic properties of the cells, like the magnitude of the ion currents for sodium, potassium, and various other currents. And it's a mystery still. It's a very big challenge. How is it that the intrinsic properties in one cell, in a bird, in a network, become the same as the intrinsic properties in the next cell? It's as if your heart rate and my heart rate become synchronized somehow. They're intrinsic to each of us, but through some network property, they become synchronized. That's a wonderful challenge for us to think about. But that's what the evidence is leading us to. So we did, let's be clear here, this is my postdoc, Arij Dow, who's now an assistant professor, manually adjusted the magnitudes of these individual ionic currents, just manual adjustment, and he was able to get models that fit the actual biological traces well and make good predictions both for other step currents and for these more complex chaotic currents that we were injecting into the cells in service of the collaboration with Henry's group. And we could show for than what our estimates were of, say, the sodium conductance or another conductance, which is called the SK current, that they showed clustering within. So here are all the cells from one bird, here are all the cells from another bird, all the cells from a third bird. They're clustered, and this is a 2D cut through that 5D space, because we looked at 5 conductances. But any given bird seems to be randomly distributed in this space. Okay. Now, manual fitting can lead to degeneracies, and so it's not necessarily a good thing, a good way to go about it. But then we used a supercomputer in a sort of brute force parameter search, and for each cell we examined 2 billion possible loci in this 5-dimensional space. And in fact, we did seem to find global minima for these neurons, and that's another result that is unexpected. And so again, the parameter estimation techniques, the more powerful parameter estimation techniques that Henry will talk about, should be able to give us insight into whether these cells really do have global minima and delve further into this complex space. But here is the kicker. Here is the kicker. What is driving the variation in this parameter space? Right? We have a given bird has one particular complement of magnitudes of sodium conductance, magnitudes of potassium conductance, magnitudes of the small current, the SK current, of the hyperpolarized, the H current, and blah, 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 blah. And all the cells for that one bird sit somewhere in that 5-dimensional space. And then all the cells from another bird sit somewhere else in the 5-dimensional space, and so on. What's driving that variation across the birds?
It's the world outside it.
It's what?
The world outside it. It's a response to the outer world, no?
Well, here's what it is. We take now the difference. We just we find the centroid for each group of cells from a bird, and we look at the distance in the 5-dimensional space, just the Euclidean distance. And now we take the songs of those two birds, and we ask how similar are they acoustically, and there are standard techniques, standard simple measures for doing that in the field. And what we find is that the two are related, which is like, give me a break. It's not supposed to be this way, but it is, right? Just as a starter, there's nothing that constrains points from being in this part of this distribution, but there are none. Okay? You can believe that it's statistically significant, okay? And it's actually significant at the level of individual animals. So there's this incredibly powerful constraint that is connecting the intrinsic properties, the distribution of intrinsic properties in this one class of cells to the acoustics of the song. All we've done so far is manually manipulate those 5 parameters, but clearly this is a wonderful opportunity to use the power of the, these more complex data assimilation procedures for asking the question about all the parameters in the cells. Okay? Now, this result makes a lot of predictions right away. Like it predicts that, well, what about birds that are singing very similar songs? They shouldn't be scattered in the space, like sibling birds. So here are 4 pairs of sibling birds. 1, 2, 3, 4. Their spike waveforms are, in fact, very similar to each other. They sit right on top of each other. How bizarre.
And these siblings were trained on the same songs?
They're singing the same songs. They have very similar genetics. They have very similar developmental trajectories. The recordings were done at about the same ages. There are all sorts of confounds with this, but at least the basic prediction is proven. Another prediction that to address that would be to look whether these properties develop, and in fact they do. So in juvenile birds, there's a lot of scatter, but it's in a small region of space, but it's only 4 birds, so I don't know if this is really going to obtain. And you even have 2 sibs. These are now 2 sibs that are juveniles, and there's lots of variation across them. So there is a developmental component to this as well. And here's the last piece of experimental data, which is good because we're all getting hungry. So we have, we have lots of tricks, right? So here's another trick. We can put an accelerometer on the bird's head. Okay, right now as I'm speaking to you, there's vibrations right? There are substrate-borne vibrations. The reason your voice sounds different to you than everyone else is you're hearing not only the airborne sound, but also the sound that's vibrating and connecting to your middle ear. So we put an accelerometer on the bird's head, and instead of measuring the song with a microphone, we measure it with the accelerometer. The advantage of that is we can then use the recording for the accelerometer to present altered feedback to the bird. So for instance, we take the song of the bird, we delay it by 50 or 100 milliseconds, and now we play it back to the bird. If I did that to you and asked you to speak out loud, I don't have to stick an accelerometer to your head, I just put headphones on you, you speak into a microphone, it'll be extremely confusing to you. And your speech will start to slow down and you'll stutter. Lots of you will do that. Lots of you will do that under those conditions. In a songbird and a zebrafinch, it takes a bit more time, 2 to 6 days, the birds actually do start to stutter. So their song changes. So we do this experiment now and ask, how do those intrinsic properties of the cells change? And what we find is, remarkably, we can actually change the shape of the waveforms of the cells in these birds by putting them in this continuous delayed auditory feedback environment. Okay? And there's a time-dependent phenomenon as well. So yeah, maybe we shouldn't give up, right? There's a real opportunity. All these features that I showed you are constraints on opportunities for building cellular models, because we know a lot about the constituents of some of these cells, especially the ones in HVC. And constraints about how the networks work. Constraints how they're connected together, to be sure, but also constraints about the sorts of behaviors that they have to produce. So I'm not ready to give up, but I am ready to have lunch. So I can stop here. If there are any questions, I'm happy to answer.